you heard, in chemical and biomolecular engineering at the University of California, Berkeley, and an investigator at the Lawrence Berkeley Laboratory, the Helen Wills Neuroscience Institute, the Chan Zuckerberg BioHub, and the Innovative Genomics Institute. She's won a plethora of awards, including the DARPA Young Investigator and the CNE News Talented 12. Professor Landry, the floor is yours.
Great. So let me just quickly go into presenter mode.
Perfect.
Perfect. Right. Just shuffle my screen just 1 second, please. All right, great. All right, thank you for the invitation to speak. Yeah, I'm very happy to be here and share some of my lab's work. So as Paul mentioned, I'm Markita Landry, an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley, where my lab is largely a nanotechnology lab. And so I thought I'd start first briefly with just a quick introduction into what my lab does on a broader level before diving into a specific area of research that we work on. And so, as I mentioned, our lab is largely a nanotechnology lab where we like to leverage the unique optical and physical properties that are conferred to nanomaterials in cases where materials are confined to the scale and quantum confinement take over. So one way that we can leverage these materials is in the development of optical probes. So for example, carbon-based materials, when confined to the nanoscale on unique optical properties, can be surface functionalized to be responsive to small molecules. And so in this way, we can develop optical probes that will respond to small molecules known as dopamine. We can then take these probes, implant them into the brain, and watch as neurons communicate with each other through these neurochemical signals. And so in this area of our research, what we like to focus on is the study of neuropsychiatric and neurodisorders, whereby communication between neurons impaired in cases in disorders. Another part of my lab also leverages the unique physical properties of nanomaterials. And for this area of our lab, we take advantage of the unique tensile strength that materials often undertake when confined to the nano, along with their high surface area to ratios. And for this area, which is what I'll talk about today, we can graft functional molecules onto these nanomaterials and use these materials to shuttle DNA, RNA, and proteins into biological systems. And the biological systems in question are going to be plants and crops. Recently focus on these areas, hopefully will become apparent throughout the talk. And so the reason that we often focus crops and plants for genetic engineering are multifold. And so, for example, we would want to genetically engineer plants, or that genetic plants can confer desirable traits to crops, such as increase to drought or pathogens, so for abiotic stresses, which is absolutely critical for our sustainability future, especially to feed a growing population amidst climate change. In pharmaceuticals, about 20% of our drugs come from, so understanding the biosynthetic pathways that generate these important small molecules and being able to upregulate these biosynthetic pathways is also quite important. And lastly, in the bioenergy sector, genetically modified plants can confer desirable traits to bioenergy crops. All of these applications depend on getting the workhorses of molecular biology, so DNA, proteins from outside the plant to inside the plant. And this is a big challenge, especially compared to other organisms because of the presence of the plant cell wall. Now, the tools that are currently used to deliver DNA, RNA, and protein to plants are depicted here. On the left, we have agrobacterium-mediated delivery. So this is a biotic method whereby a pathogenic agrobacterium is genetically engineered at the bench to contain a plasmid of interest. This agrobacterium can then infect plant cells, thereby transferring DNA and enabling DNA integration into the plant host genome. Agro-mediated delivery is extremely efficient. However, there is a range limitation. Because this is a biotic method, this method only works for certain types of plants and used for wild crops and a lot of plants that we would have interest in genetically manipulating outside the lab. Furthermore, agro-mediated delivery can only deliver DNA. Conversely, biolistic particle delivery is accomplished by dehydrating particles, DNA, RNA, and protein cargos onto micron-sized gold particles. These particles are then loaded onto a gene gun and biolistically delivered into plant tissues. These delivery methods work by rupturing the plant cell wall and cell membranes and allowing integration of the transgene if it is delivered into the plant host genome. As you can imagine, this method, while more species independent, does cause quite a bit of cell damage and has low transformation efficiency, making it difficult to regenerate transformed crops. In both of these cases, there's a lack of control over the location and frequency of transgene integration, causing potential disruption of endogenous pathways. And also from a regulatory standpoint, the use of any biotic pathogen, such as agrobacterium, triggers regulatory oversight over the transformed crop as a genetically modified organism. So there's significant interest in developing technologies that are pathogen and force independent to get better control over where genetic manipulation happens in plant genomes. Despite these limitations, there are several success stories, especially in the United States, who is a global leader in the production of genetically modified crops listed here. And also some nice international examples, such as the production of golden rice, which is a strain of rice that has had the biosynthetic pathway for vitamin A engineered into it. This strain of rice glows yellow as it produces vitamin A, and the concept here being that this varietal of rice could be distributed to vitamin A deficient populations. Again, despite the advantages and the progress made in genetically modifying crops, the average genetically modified crop today takes on average 13 years of research and development at a cost of nearly $140 million to produce. And a lot of this time and money is on the regulatory side, not just on the research and development side. So as I alluded to before, the development of a technology that is species independent, but also non-pathogenic and force independent would allow us greater control over genetic manipulation of plant genomes in a manner that might be more amenable to regulatory oversight over those transformed crops. And so today I'm going to talk about two of the platforms that my lab has worked on. The first, a DNA delivery platform whereby DNA plasmids can be delivered for expression of a genetic transgenic protein. And second, a gene silencing platform where small interfering RNA can be delivered to a plant to have the opposite effect, which is to knock down levels of a protein. And I'll point to some nuances between these two different platforms, highlighting the importance not only of the carrier, the attachment point, but also the surface chemistry in being able to accomplish these two seemingly very similar goals. So first, it's important to consider what the membrane barriers are. So for example, what is possible to deliver across plant membranes? So if we think about most cells, the dominant barrier there is going to be the cell membrane. So this lipid bilayer that excludes and controls which molecules can cross. And certainly this is true for most mammalian cell systems with a size exclusion limit of about 500 or so nanometers. However, unique to plants is the plant cell wall. And the cell wall is a barrier that has a size exclusion limit of about 5 to 20 nanometers. This is about as large as a particle can be before it can no longer cross that membrane. So clearly, the plant cell becomes the dominant barrier to biomolecule delivery and must be considered in order to be able to deliver these cargoes in a force-independent and pathogen-independent manner. So then we turn to dimensionality of nanomaterials. Here I'm showing carbon-based materials as an example, but really any base is biocompatible, and certainly we've also worked with cellulose and gold-based nanoparticles as well. But for purposes of illustration, I'll show here today just our results with carbon, where we can consider dimensionalities of carbon on 2 dimensions and ask ourselves the question, what form factors would be most important? So for example, 0-dimensional nanos are small enough to fit through the plant cell wall, at least in theory. However, there's not a very high surface area to volume ratio onto which we can do chemistry or graph biomolecules. Conversely, 2-dimensional materials, atomically thin graphene, have a very high surface area to volume ratio, but no longer are below the size exclusion limit. We largely put these high aspect ratio 1-dimensional nanomaterials because they are at least in 1 dimension small enough to fit through the plant cell wall, have an extruded first dimension that gives them a high surface area to volume ratio for chemistry and for cargo loading. Now, the platform that I'll first be talking about is our DNA delivery platform. The concept here is that these high aspect ratio carbon materials would be able to load a functional molecule such as a plasmid, translocate through the cell wall in the plasma membrane, and then make this cargo accessible in this case in the nucleus, where the DNA would be transcribed and translated into a functional protein. Now, the protein we're going to be testing for expression is green fluorescent protein, or GFP, initially just to serve as a reporter for successful expression and functionality of this platform. Now, one question with the development of this platform is how tightly does the cargo have to be attached to the carbon nanotube? Because if it's too tightly attached, it might not be available for transcription. But if it's too loosely attached, it might die prematurely before making it into the cell, especially while the small cell wall. And so to answer this question, we developed 2 different chemistries that have different attachment strengths between the plasmid and the carbon nanotube. In this chemistry that is accomplished through slow dialysis exchange of the surfactant for a plasmid, we can load plasmids onto carbon nanotubes through pi-pi stacking interactions between the aromatic bases of the carbon nanotube, or of the DNA, and the sp2 hybridized carbon lattice of the carbon nanotube. So these are loaded through associations that are pi-pi stacking, so akin in energy to about a hydrogen bond, having a large dissociation constant between the plasmid and the carbon nanotube. On this second chemistry that we developed, we first carboxylate the surface of the carbon nanotube, and then through an EDC-NHS reaction, we can attach a cationic polymer, such as a branched polyethyleneamine, to these carboxyl handles. These PEI functionalized carbon nanotubes now carry a positive charge that can load DNA plasmids through electrostatic interactions between the positive carbon nanotube and the negatively charged DNA. These electrostatic grafting interactions have a high affinity, a much stronger bond than pi-pi stacking interactions. So plasmids are loaded tightly and have a low dissociation constant. We can then take these constructs and introduce them into mature plant cells to test to see if they are able to deliver this GFP plasmid and enable GFP expression in tissues. So what we do is we generate colloids that are, again, suspensions of carbon nanotubes with these plasmids, either PEI plasmids or these pi-pi loaded plasmids, and infiltrate them at parts per million concentrations into leaf tissues. We can wait a few days for internalization, protein transcription, and translation to occur before quantifying GFP expression microscopically and on a molecular level. And so here I'm showing some of our results in Benthy, which is a model plant, arugula, wheat, and cotton, which are crops. And as you can see here, we observe GFP expression at levels that are about half as efficient as agro-mediated transformation quantified by qPCR. Now, what's interesting is if we look at the effect of chemistry, comparing just dialysis to electrostatic grafting of our carbon nanotubes to our plasmids, what we see is that the dialysis method, which has a high dissociation between the plasmid and the carbon nanotube, does not generate very much GFP expression compared to the electrostatically loaded plasmid. And so what these results highlight is the importance of the plasmid being loaded onto the carbon nanotube with a high affinity for effective expression of the transgenic protein. And indeed, I'm not showing this data, but some of our recent explorations of these constructs suggest that the DNA does not even have to desorb from the carbon nanotube for it to be transcribed into a functional protein. And to give you a sense of the relative size scales that we're talking about, I've depicted here roughly to scale a plant cell, where by far the largest organelle is a large central vacuole, and some examples of previous particles that have been used for biolytic delivery. Compared to the inner diameter of a carbon nanotube, these are much larger particles. And so hopefully the schematic can help you appreciate perhaps some of the mechanisms by which these single wall carbon nanotubes are able to force independently enter plant cells. Now, one of the interesting features of this platform as we were quantifying is in the transit of the expression. So at day 3, we would observe green fluorescence in our tissues that would usually sub day 10. When we extract mRNA from these plant tissues and perform a quantitative or qPCR to measure the amount of GFP mRNA fold change, we observe the same pattern with a peak GFP mRNA fold change at day 3 that goes back to baseline at day 10. What these results suggest is that there's a lack of integration of the transgene in the plant host genome, which could be, in fact, beneficial for greater, again, control over transgene expression in plant tissues. This is in contrast to agrobacterium-mediated delivery of the same GFP transgene. Both at day 3 and at day 10, we observe strong constitutive expression of GFP, confirming integration of the transgene. And so to validate this at a higher level of sensitivity, we performed a digital droplet PCR assay. So this is an assay that we want to perform to confirm that there's a lack of transgene integration with greater sensitivity than qPCR can afford. So the experiment looks something like this. We treat leaves either with agrobacterium-mediated delivery of GFP or carbon nanotube-mediated delivery of GFP plasmids. We can confirm that there's expression at day 3, but then wait 14 days before digesting the treated leaf tissues and extracting their genomic DNA. This genomic DNA can then be sheared into fragments, and these fragments can then be partitioned into tens of thousands of nanoliter scale or nanoliter sized droplets that can then individually be assayed for the presence or absence of the transgene. We can do this for the transgene, which is GFP, and also for a control reference channel, which in our case is going to be elongation factor 1, which should not change as a function of treatment. And so this is what the data looks like for agrobacterium-mediated treated leaves at day 14. We see plenty of positive hits in the control channel as expected, and we also see many droplet hits in the GFP channel, confirming integration of the transgene when the transgene is delivered with agrobacterium. However, at day 14, despite seeing expression at day 3, we see plenty of droplet hits in our control channel, but none in the GFP channel, showing with a greater degree of sensitivity that there is a lack of transgene integration of these plasmids when delivered with carbon nanotubes. And so, given the transience of the expression of our platform beyond expressive port approach, we're excited to move forward into addressing the ability to genetically manipulate plants for the reasons that I motivated at the beginning of the talk, both in terms of identifying but also modifying these genes of practical relevance beyond expression of fluorescent protein reporters. And specifically what we have in mind is CRISPR. So CRISPR is a genome editing technology that can be coded for in a plasmid plants. The concept here is that the CRISPR is able to be expressed and would enable gene knockout or gene knock-in a manner void of integration of the CRISPR machinery itself into the plant host genome. And so this is a concept that would allow the expression of Cas9 as a protein along with its guide RNA complexation of which would produce an protein complex that could then search the genome of the plant to perform a site-specific edit. That edit would, of course, be conferred permanently and could perhaps be regenerated into transgenic plants by avoiding the integration of the CRISPR DNA itself into the plant host genome as well. And this is a technology that could be especially useful for crops that are vegetatively propagated, for which transgene segregation through breeding is not possible, such as cassava, banana, and sugarcane. And so towards this end, some preliminary studies in my lab have looked at optimizing the CRISPR machinery complex in plants delivered with carbon nanotubes. And the experiment I'm showing here is one in which we are taking transgenic Cas9 expressing Nicotiana benthamina plants and introducing guide RNA that is coded for with a plasmid delivered by polyethylenamine coated carbon nanotubes. And so here, the guide RNA would allow the CRISPR, or would allow the Cas9 that's being constitutively expressed in these leaves to form an active RNP and to perform a site-specific edit at PDS, a pigmentation site in these Nicotiana benthamina leaves. We can do this with agro-mediated delivery of the same CRISPR plasmid or the same guide RNA plasmid. And as you can see here, when delivered with carbon nanotubes, we're a little bit under half as efficient as when we are delivering these CRISPR plasmids with agro. However, these results are still promising because, as I mentioned before, agro-mediated delivery will perform or will enable constitutive expression of this RNP complex. So the dose that we're getting with agro is much higher than the single dose we're getting with transient expression of our carbon nanotubes. And so in moving forward, we're excited to try, for example, multiple infiltrations that would allow us to increase our editing percentages with PEI delivery of these guide RNA plasmids. We're also, of course, interested in moving into other tissues that can be regenerated into whole plants, including reproductive organs of the plant and also plant callus tissue, such that the transformed tissues could be, again, regenerated into edited crops, perhaps avoiding, again, segregation-based, breeding-based segregation of integration of the transgene. Now, shifting gears a little bit and talking about a different application of relevance, the delivery of small interfering RNA into plants is also an interesting concept that would allow us greater control over probing biosynthetic pathways in leaves, and is also, RNAi is also an interesting potential alternative to the use of chemical pesticides in plants. And so for these reasons, we wanted to see if some of the carbon nanotube concepts we developed in DNA delivery would also for the delivery of small interfering RNAs. And of course, what we first tried was the best performing construct from our DNA delivery studies. So if you remember, these PEI carbon nanotubes worked really well to deliver GFP DNA vectors. And so what we did is we took small interfering RNA that knocks down green fluorescent protein, and we tried to deliver the small interfering RNA to transgenic GFP benthy plants. So these are tobacco plants that constitutively express green fluorescent protein, and our goal here is to knock down that green fluorescent protein with the delivery of this siRNA. However, when we infiltrated these constructs into our GFP plants, we did not observe any silencing of our GFP inside the plant tissue. And again, this was surprising to us because this is the construct that had worked so well for DNA delivery. We thought a little bit more about the chemistry and the motivation for developing chemistries of varying dissociation constants, and thought that since this RNA construct is relatively small and needs to be accessed by the RISC protein complex inside the plant for silencing, perhaps a looser association between RNA and carbon nanotubes was needed for this particular application. And so we performed a few thermodynamic calculations that suggest that this is indeed the case. So if instead of using electrostatic interactions, we instead use pi-pi stacking interactions to separately load the sense and anti-sense strand of the functional RNA onto two separate carbon nanotubes, we can calculate that these constructs will be stable in the pristine extracellular environment, for example, in a buffer solution, where the preference is going to be for the RNA to remain on the carbon nanotube surface. Once introduced inside the crowded molecular environment of the plant cell, here it would be thermodynamically advantageous for ligand exchange to occur, protein corona formation to occur, and desorption of the siRNA individual strands to an active siRNA duplex, especially if these two strands are complementary to each other, which they are. And so by instead loading the siRNA in this manner and injecting it or infiltrating it into our Nicotiana GFP plant leaves, what we now see is strong silencing of the GFP transgene through introduction of this siRNA. So we can confirm this both on a microscopy level, but also by extracting all proteins from these tissues and performing a Western blot to quantify the amount of GFP in these tissues. We can also perform controls, for example, in the delivery of RNA that does not target GFP, where we don't observe any silencing. If we introduce siRNA that does not target GFP, we see no silencing. If we introduce siRNA that does target GFP without carbon nanotubes, we see some degree of silencing, but much greater silencing if the RNA is delivered with carbon nanotubes. And this is quantified with a qPCR, and this is quantified with a Western blot. So here we're quantifying mRNA, here we're quantifying GFP protein. Now, in addition to enabling RNA to go inside the cell, another potential mechanism that allows high efficiency could be the protection of these cargoes from degradation. So in the case of DNA, but especially in the case of RNA, these are polymers that are highly susceptible to endonuclease degradation. And so what we did is we performed a single molecule total internal reflection fluorescence microscopy assay. And this is a technique that allows us to assay the integrity of DNA or RNA polymers on a single molecule level. The assay works something like this. So we can take the RNA strands in their single-stranded fashion that are, again, going to be targeting GFP knockdown and label them with a Cy3 dye. We can then take these individual strands of Cy3 labeled RNA and put them on the surface of a microscope slide. What we can then do is treat this slide with RNase A for 5 minutes and then quantify the amount of fluorescent spots that are decreased post-RNase treatment that suggest degradation of those spots. And as you can see here, following RNase treatment, we see almost complete degradation of the RNA. However, if we take this fluorescently labeled RNA and first load it onto a carbon nanotube and then immobilize that construct and treat it to RNase A, we do see some degradation of fluorescent spots or some elimination of fluorescent spots, but nowhere near as much degradation of the RNA as we did in its free counterpart. And so these results suggest that in addition to helping shuttle these cargoes inside plant cells, another mechanism by which these nanoparticles could be helping gene expression or RNA-based siRNA silencing is in the protection of those cargoes until they are able to be functional inside the plant cell. So lastly, to wrap up the talk with a question that we've often asked ourselves is, if we're going to be developing these technologies for plants in a higher efficiency manner and for much more difficult cargoes such as proteins, we need to know why it is that transport across the cell wall is even possible. And so this is a question that a former postdoc in my lab, Huang Zhang, undertook. Specifically, how do nanoparticles and their parameters affect transport across the cell wall? And for this particular area of study, we switched from using carbon nanotubes and instead used DNA nanostructure nanotechnology and also DNA origami nanotechnology. And the reason we made this switch for this particular question is because DNA nanostructures provide really great control over the generation of nanoparticles that have controllable shape, size, and rigidity in a way that's difficult to do with carbon nanotubes or most other nanoparticles for that matter. Now, the way that this technology works is that we can take a collection of staple strands in the case of 3D nanostructures or DNA and staple strands in the case of DNA origami nanotechnology. And because these are DNA bases, we know the interactions between these staple strands amongst themselves and or with the DNA backbone. So we can programmatically predetermine what shapes, sizes, and rigidities of structures we can make through the thermodynamic annealing of these ligands to make three-dimensional shapes. And others have made really small Mona Lisas and smiley faces. In our case, what we made were a library of different shapes to test the effect of shape on nanoparticle internalization. So for example, we made a 2.4 nanometer DNA nanostructure tetrahedra. We made 5 by 16 nanometer hairpin tile structures, hairpin assemble onto nano strings. And we can also, because these are DNA-based, introduce sites loci onto these structures on we can attach DNA, RNA, or protein cargoes. So depicted by red arrows here, we attached a protein cargo, here neutrophin protein, onto the vertex of the tetrahedron, onto the 10 monomeric units of the nanostring, or on center versus the side of these hairpin tiles. And we can confirm with high resolution atomic force microscopy that we are indeed attaching these protein cargoes to the loci that we predict. Now, the first, or the experiments that we're going to be doing here are testing for internalization. And this is somewhat difficult in plants because, as I mentioned before, one of the largest intracellular organelles in plants is the plant cell vacuole. So if we look at plant epidermal leaf cells, which is what we're looking at here, in a transgenic GFP-expressed Nicotiana benthamiana plant, what we'll see are these jigsaw puzzle shapes. Now, GFP is being expressed into the cytosol of these plants. And so what you're actually seeing here are plant cells with a large central vacuole that is pushing the cytosol out to the side or the periphery of the cell. So the GFP is really being used in our case here as a cytosol marker to tell us where the cytosol is in these micrographs. We can then also fluorescently label our DNA nanostructures with 5,3 dye, inject those into the plant tissue, and then through a colocalization analysis, look at the relative levels of internalization as a function of some parameter by calculating the colocalization fraction between the 5,3 nanostructure channel and the cytoplasmic GFP channel. And so if we look at the effect of internalization on something like size, for example, we'll see that as a function of size, 2.4, 8.8 versus 12.6, we see a much higher colocalization fraction for the smaller nanoparticle than the larger nanoparticle. And these results are as we would expect. As particles get larger, it becomes more and more difficult for them to internalize into the plant cell cytosol. Now to look at the effect of shape with the same assay that I just described. And so here our hypothesis is that the nanostring would actually internalize best because it is most similar in form factor to these high aspect ratio carbon nanotubes that we use that have worked so well for DNA and RNA delivery. However, it was surprising to us to see that in terms of colocalization fraction, these nanostrings actually internalize less than the tetrahedra and hairpin tiles, again, as assessed through this colocalization assay. And so we were perplexed by these results, and what we did to test the effect of other parameters was to take this nanostring and we tethered it to a carbon nanotube. And the reason we did this is because we know that the carbon nanotubes internalize. We know that the nanostrings don't internalize very well. So what would happen if we tethered both of those together and then put them into the plant tissue? What we see is that carbon nanotubes, as I mentioned before, internalize quite well as measured by colocalization analysis. Nanostrings largely do not. And the nanostring plus nanotube complex internalizes just as much as carbon nanotubes. And these are interesting results because it suggests that, of course, this complex is much larger than either construct by itself. But the main difference between nanostrings and carbon nanotubes is in their tensile strength, where carbon nanotubes are several orders of magnitude stiffer than our nanostrings, suggesting that stiffer or more compact nanoparticles are also a feature of merit that increase the internalization of those particles across the cell wall. And these results also probably explain why free DNA plasmids don't just go through the cell wall by themselves. They need a carrier that's much higher tensile strength to shuttle them across in our previous DNA studies. Now, because these nanostructures, again, have these loci onto which we can attach functional cargoes such as siRNA, we decided to next test if these nanostructures could deliver functional cargo plants. So the assay that we're doing here is we're going to be tethering small interfering RNA to our nanostructures, infiltrating them into our GFP, and seeing if we knock down expression of GFP. So as you can see here, these different nanostructures post-treatment to these Nicotiana benthamiana leaves, we see a decrease in fluorescence intensity, suggesting a higher degree of silencing, especially with these three constructs, which matches the level to which they internalize in these leaves. We can, again, molecular level, validate this by digesting the plant tissue, extracting all proteins from the plant tissue and quantifying on a Western blot the amount of GFP that's expressed post-treatment. Now, what's interesting is that here I'm reproducing the results from our previous slide, showing a decrease in GFP expression as quantified with a Western blot. So we're quantifying the amount of protein that is decreased following treatment with siRNA-loaded nanostructures. If we perform a qPCR that measures the messenger RNA specific to expression, we would expect a decrease, a commensurate decrease for those structures as well. However, what we have a very different trend where we only see the predicted decrease in GFP-specific mRNA foliage when RNA is delivered freely or when this RNA is delivered to the vertex of a tetrapod. We instead see increases in GFP-specific mRNA when siRNA is delivered on either position in a hairpin or on nanostring, which despite seeing protein level silencing. And so the ongoing hypothesis for what this might be is in consideration of what makes these two structures different from these three structures. And the main thing that I can think of is in their steric accessibility. Free RNA or RNA on the vertex of a tetrapod is much more sterically accessible to the RISC protein complex that would be needed to silence at the protein level, whereas RNA delivered through these other three structures is sterically inaccessible. So though we haven't shown this mechanistically, ongoing hypothesis is that sterically accessible siRNA will undergo canonical transcriptional gene silencing through cleavage of the GFP-specific mRNA, whereas sterically inaccessible siRNA delivered on these nanostructures or perhaps other nanoparticles might instead undergo post-transcriptional gene silencing through translational repression, but without cleavage of the GFP-specific mRNA, causing an accumulation here, but causing a clear degree of silencing at the protein level. And so these results suggest the importance not only of the efficiency of the platform in terms of getting the cargo in, allowing the cargo to be functionally used inside the plant and not degraded against endonucleases. Lastly, that the way that we attach the cargo onto the particle also have disparate effects on how the function, in this case silencing, is accomplished. And in a manner also that is specific to how those cargoes are going to be used by the endogenous machinery of the plant tissue. And so we'll wrap up with this one slide showing some ongoing studies in studying these transport properties in plant tissues, specifically looking at the effect of something like shape on the ability for these particles to enter plant cells. So here I'm comparing gold nanoparticles, specifically 15 nanometer versus 13 nanometer gold nanospheres and rods, where the main difference here is again in the aspect ratio of the gold nanorod. Labeling these with Psi3, introducing them into plant tissues, and now asking the question, how long does it take for these particles to internalize? What's interesting about these results is that despite, again, the aspect ratio being the main difference between these two particles, we see very different kinetics of internalization into plant cell cytosol. So for example, these gold nanospheres internalize quite slowly with an apparent peak internalization 12 hours post-infiltration. Conversely, gold nanorods seem to have a very sudden internalization that peaks at around 6 hours post-infiltration, so much faster than the gold nanospheres, again, despite the aspect ratio being the main difference here. So some ongoing modeling studies in my lab to help understand these phenomena, and again, with the motivation of being able to understand these properties so that we can develop better technologies in the future. And so with that, I'll wrap up the talk just by briefly outlining what I've talked about, specifically DNA delivery, where we've shown that DNA plasmids can be delivered and transiently expressed without transgene integration into plant tissues. And many of our ongoing and future studies will involve the delivery of CRISPR-Cas endonucleases for transient expression, but permanent editing of the plant host genome, and perhaps using some of the other technologies that we've developed, such as our gene silencing technologies for RNA delivery to deliver other DNA and RNA types, such as donor DNA templates, small synthetic guide RNAs, and perhaps even messenger RNAs for DNA-free technologies altogether. I've also alluded to the importance of understanding these transport properties based on nanoparticle physical and chemical properties, work that we've done with DNA nanostructures, and we hope to do in the future with gold as well. So with that, I would greatly like to thank the slew of collaborators that we've had for this work, specifically Brian Saskowitz and the support of the IGI for the early stages of this project. in addition to mentioning the work that's been done largely by Guzda Demerer, Juan Zhang, and Nat Go in the lab, and Frankie Cunningham, who's also working on genome editing technologies in the future. For the trainees in the audience, I would also like to mention that Guzda Demerer, who led the work that I talked about today, will be starting her lab at Caltech in July of 2022. So if you're interested in performing this line of research, I highly recommend you reach out to Guzda for training opportunities. And though I didn't talk about any of our neuroimaging studies, I would also like to mention a recent graduate student of mine, Abraham Billene, started his lab a few months ago at the Genelia Research Campus in Bethesda, and is also looking to hire trainees into his lab. So if you're interested in plant genetic biotechnologies, nanotechnologies, and in brain imaging with semiconducting nanoparticles, please reach out to Guzda and Abraham for opportunities in their respective labs. And with that, I would like to thank you for your attention and glad to answer any questions.
Thank you so much. Let me turn my screen back on here. Oh, beautiful talk. And we have a number of questions from the audience. I can start my video. There we go. So the first one is, it's advancing. There we go. Using CRISPR in plants, very interesting. Will it cause problems like species invasion? Do you have to worry about the genetically modified plants?
That's a great question. Yes, and I think, you know, in terms of species invasion and lack of species diversity, that's a track that we went down with the agricultural revolution, right? So we're very much already, you know, facing some challenges that are the byproducts of modern agriculture, and I think that those are important to consider. That are independent of nanotechnology, independent of CRISPR, independent of the specific breeding technologies or the specific molecular technologies that we use to get these crops. So that's, I think, one separate consideration. I do think that technologies such as CRISPR, given that they can be much more precise than conventional non-GM approaches, are going to be net beneficial, because there we're just changing fewer genes. We're manipulating the DNA less than we do with conventional non-GM technologies. So given that we need to be in the agriculture kind of practices that we are in now, I do think that CRISPR is one of our best bets in moving forward.
And our next question is, are there lessons learned from plants that can be applied to gene editing in human or other animal cells?
That's a good question. Yeah, and I think you know, plant genomes are much more, surprisingly, much more complex than ours, right? So plants have very large, very complex genomes, very few of which are sequenced, even fewer of which are annotated. So it's quite difficult, I would say, to do a lot of these studies in plants for that reason. So if anything, I think that editing in human or other animal cells has taught us what we know in the plant sector about what works and what doesn't work, both from the introduction of molecular biology tools in these cells, but also in terms of what's needed, the complexity of information that will be needed to get plants up to speed with animal cells. I think certainly from a kind of bioethics standpoint, genome editing in plants poses fewer potential risks than editing in human or other animal cells. So this is, you know, more my personal opinion, but I do think that the more immediate benefits of CRISPR would probably be in the agriculture sector versus in human editing.
And is there, because there's less of a toxicology concern, does that open up materials to you in a useful way to follow up on that?
Yeah, certainly. So biocompatibility is important in any sort of biological system, but especially so if it will be introduced into a human. In the agriculture sector, where I like to draw the line on the use of these materials is in their degradation. So for example, carbon nanotubes largely do not degrade, at least not on any time scale that we have done any substantial studies on their impacts in bioaccumulation in the environment, et cetera. So I would advocate for the use of tools such as carbon nanotubes only in a molecular biology sense, where they can be used to introduce a transgene, but then where any sort of plant product that would come from this type of genetic manipulation would be many generations down the line, devoid of direct exposure to the nanomaterial. If we wanted to do RNAi, for example, in a field, introducing RNA to plants in the wild, there we would, at the very least, need to work with materials that degrade into components that are quantifiable and that will not pose any bioaccumulation issues. For some of our RNA delivery work, that's where we are interested in heading, is in development of biocompatible materials such that that can be, far down the line a possibility.
Fantastic. And then Professor Markita, using nanoengineering and plant DNA editing, is it possible to do something like optogenetics in plants?
Yeah, so I think that maybe it was 2 years ago, there was a really interesting study published using optogenetics to control plant stomata. Plant stomata are the plant's breathing organs. They open and close for gas exchange. And it was shown that optogenetics could be used to help these stomata open and close to increase respiration and showed perhaps, I believe it was increase in photosynthesis or something of that nature. So it is possible to do optogenetics really in any genetically tractable organism that you can introduce channel rhodopsin into. Of course, it is most heavily used in the brain, but it's certainly possible to introduce channel rhodopsin into plants as well.
Mm-hmm. Terrific. Well, thank you so much. As I said earlier today, normally we would walk across the stage and bring you a plaque from ACS Nano and iCANX. The best that we can do at the moment is this electronic certificate, and to thank you for this absolutely beautiful work and talk. And we're looking forward to much more from you in your laboratory, as well as your wisdom at the journal and the forward-looking piece that you'll put together for us in any of the different areas that you work. We'll all be looking forward to reading that. Thank you to Professor Landry and to our other two speakers, Professors Tuo and Chen, for this fantastic ACS Nano Rising Star set of talks, the last one of 2020, but we'll look forward to more in 2021. Next week, we'll have Mine Young Scientists on December 11th. On December 18th, we'll have a talk show panel about what we're trying to do with the iCANX lectures and series and connecting the world together. So stay tuned for that. And in the meantime, our global community continues to grow. And I want to thank our speakers today one more time.
Thank you.
Thank you.